Okay, the second macromolecule that we need to discuss are the lipids. And in your diet, you commonly refer to these substances as fat, and they do include animal fats and also oils, the cooking oils that you use, um, both solids and liquids. Butter is an example of a lipid, and canola oil or sunflower oil, those are also lipids. And then also the waxes, and you see these if you get a really shiny apple, that's because of the wax coating on the outside of the peel. These compounds are not soluble in water, so they're hydrophobic and they repel water. And that's one of the reasons why you find a waxy coating on the apple is it forms a protective coating that doesn't allow water to exit the cells in the apple, to prevent dehydration. And in a lot of your house plants, some of them have waxy coatings on the leaves for that same reason. You can identify a lipid by looking to see what atoms it's composed of, and when you look, you should only see the C's, H's, and O's, the same as you saw in the carbohydrates. But this time the numbers will be different, and they will not be in that 1 to 2 to 1 ratio like the sugars. Lipids are polymers. They're made up of smaller building blocks, and in this case, lipids are kind of special compared to the other three polymers because lipids are made up of two different types of building blocks. You have fatty acids that are combined with a glycerol molecule, and that's what builds a lipid. So in these diagrams, you can see the different monomers joined together. And so the black oval is circling the glycerol molecule. So you have a glycerol molecule here, and you can see the backbone or the chain of glycerol here in this one as well. And then both of these are triglycerides because they have 1, 2, 3 fatty acid tails. This one also has 1, 2, 3 fatty acid tails. And so this long chain of C's and H's, carbons and hydrogens, attached to the glycerol molecule, this is the fatty acid portion. And there's a difference between these two lipids. One is going to be a saturated fat, and the other one is going to be an unsaturated fat. So how do I know that? How do I tell the difference between a saturated and unsaturated fat? It also comes down to their chemical structure. But if I can't see at the molecular level, and I'm just dealing with what I can see, then saturated fats are going to be solid at room temperature, like this stick of butter. And then the unsaturated fats tend to be liquid at room temperature, like your vegetable oil. The function of lipids, we use them to store energy. These are more long-term energy molecules because it's not so easy for our cells to quickly break them down and release the energy. And then, as I was saying before, they can be a form of waterproofing for plants and animals. This slide points out that some feathers have waterproof covering on them in the form of a lipid or wax. Lipids are also an important feature of the cell membrane. Every cell in your body is surrounded by a double layer of lipids, a bilayer of lipids that prevent water from escaping the inside of the cell, because lipids are hydrophobic, so they're going to keep water from going out of the cell. We also use lipids to cushion and insulate our internal organs. So it would not be good for you to not have any body fat. It would be a dangerous situation. So you definitely need to have some body fat. Also, lipids surround your neurons in your nervous system and provide insulation and a kind of a conductor capacity to your neurons. I'm going to go in reverse here for a second just to show you the difference between, I think we skipped over this somehow, it's saturated and unsaturated fat. An unsaturated fat, you will be able to see at least one double bond. So I'm looking right here at the kink in this fatty acid tail. And that kink is because of this double bond orientation making a bend in the last part of the fatty acid. So looking over here, I don't see any double bonds. There are no double lines between two carbon atoms over here. So this one is completely saturated with hydrogens. If we were to break this double bond, and then there would be an opportunity for two more hydrogen atoms to attach here, but that's not the case. So we say that this one is unsaturated because there's potential for more hydrogen to bond. But this one over here is saturated. There's no double bond, so it's at maximum capacity for hydrogen. So if you want to tell the difference between those two, you simply look for a double bond. And you can see that this one is also an unsaturated fat because there's a double bond here, 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 and here. Okay, now moving to the nucleic acids. This is either DNA or RNA. There are only two kinds of nucleic acids. And you can identify these because they will contain carbon, hydrogen, and oxygen, and then in addition to that, nitrogen and phosphorus. Nucleic acids are polymers. The monomers that get joined together to make them are called nucleotides. So you have repeating combinations of nucleotides that build a nucleic acid. And the function of nucleic acids is to store and transmit genetic information. That one you're probably familiar with. The last class of macromolecules are the proteins. And proteins are essential to the inner workings of your body and your cells. And you can identify a protein because they will contain carbon, hydrogen, oxygen, and then in addition to that, nitrogen and also sometimes sulfur. So the S comes in here with proteins. The building blocks or the monomers of proteins are called amino acids, and there are 20 different amino acids. And by combining different arrangements of those 20 different amino acids, we can get literally an unlimited number of different proteins. All of them have a pretty specific function in your body. The 20 different amino acids you can recognize by their structure as well. Each amino acid will have four basic parts. A carboxyl group, which this circle here is, you're looking at a carboxyl group. It has a carbon double bound to an oxygen and then another oxygen attached here. Carboxyl groups have a negative charge. All amino acids will have an amino group, and this circle has an amino in it that's usually associated with a nitrogen atom attached to hydrogen, so NH3 or NH2. Those are amines, different types of amino groups. And then all amino acids also have just one single hydrogen attached to the central carbon. So on all of these are four different types of amino acids. They all have that central carbon. They all have a lone hydrogen. They all have an amine group or an amino group here, 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 and here. And they all have the carboxyl group. You can see that on all four. So all four of these have those three parts in common. The only difference, and this is what designates one amino acid from a different amino acid, are the R groups, or sometimes referred to as a functional group. And so they'll have a little different grouping or smaller unit as that fourth bond on the carbon, and that will indicate what amino acid that you are looking at. There are many, many different functions of proteins in your body. The enzymes that catalyze your chemical reactions. Enzymes are just a special class of proteins. The hormones, which you're a teenager, you're probably pretty familiar with hormones and how they work in your body. Hormones are all proteins. You have proteins in your body that are used, they attach onto other things and transport them in the body, such as hemoglobin that's found in your red blood cells that carries oxygen. That is a protein. Your muscle tissue, you're probably most familiar with this being a protein, but that's what causes your muscles to contract. In your immune system, it recognizes antibodies on different antigens in your body, and those antibodies are proteins. There are proteins that extend across the cell membrane that help in recognition and also help transport things in and out of the cell. And then there are structural proteins that you find in your bones and muscles that give you support. Finally, let's discuss a little bit more specifically the enzymes. Enzymes are all proteins, as I just said, and we've already talked a few times this year that enzymes are used to speed up chemical reactions in your cell. They don't cause the reaction to happen. If you gave those reactants enough time, the reaction would eventually occur, but sometimes they would occur so slowly that you would die before they happen. Enzymes are very specific to the reactants that they are interacting with, and so they will only carry out, you have a specific enzyme for a specific chemical reaction, and then they do that job extremely well. You don't use up your enzymes, so they're reusable. You can recycle your enzymes and use them over and over again. Taking a look at how they work, the purple glob here is the enzyme itself, and in the enzyme, because of the chemical structure, the way that the carbon atoms are arranged and the amino groups are attached to each other, it forms a pretty specific active site. This is where the reactant is going to bind. And so this active site is kind of like a lock. It's a specially designed pocket where only a certain shape of key will fit into it. And so it's designed specifically with those keys in mind. And chemically, we call those keys that fit into it the substrate molecules. So the substrate and the other, these would be the reactants in the chemical reaction. The enzyme simply locks them down into place and causes them to react. Once the product of the reaction is formed, then they leave the active site and it becomes available to catalyze the next set of substrates, and the reactions just keep on going. The intermediary here that you're looking at, when the substrates are in the active site, we call those the enzyme substrate complex. So that's this entire entity. And then, you release those and just have the enzyme again. So remember that the shape of the enzyme, so that goes back to the chain of amino acids being very specific for that specific enzyme. It gives it a specific shape that only fits one certain type of substrate, and that's what maintains the control that your body has over regulating that chemical reaction, is that you only make enough enzyme that you need to catalyze that reaction, and that enzyme only works on that specific reaction. We know that there are over 2,000 different types of enzymes in the human body system that are working to keep you alive. And then just also remember that they are reusable. So once the substrate forms product, the product leaves, and then the enzyme can be used again for the same chemical reaction.